Right, I think we're gonna start. Hello, everyone. Welcome to today's webinar. We are honored to have Professor Stuart Baker, who is a professor of movement neuroscience at Newcastle University, UK, who will be talking to us today about portable neuroscience using devices and apps for diagnosis and treatment of neurological diseases. I'll give you a brief bio of Prof. Stuart Baker before we go on to the talk. So he studied natural sciences in Cambridge and then went on to do a PhD in neuroscience in Cambridge and London. From PhD onwards, his work has involved studying the neural control of movement, especially the primate hand. He makes neural recordings from macaque monkeys, and his laboratory is quite unusual because they do not just study the cortex, but they have developed methods also to record from brainstem and spinal cord. He also uses non-invasive methods in human volunteers, such as transcranial magnetic brain stimulation, which allows fruitful interplay between animal and human measurements. His recent work has focused on the role of reticulospinal tract in primate motor control. And as you can see from the talk title, Portable Neuroscience Using Devices and Apps for Diagnosis and Treatment of Neurological Disorders, I think this has so much relevance to the African setting where we are still far behind in terms of making diagnoses and the lack of tools for making accurate diagnosis of dementia and other neurological disorders. So it is hoped that hopefully from this conversation, there will be some interest as well as collaborative opportunities to see how that can be carried along in terms of research in the African continent. So for those that are on Facebook, you can type your questions, which will be answered at the end of the talk. Hopefully, I'll go to Facebook as well as through Zoom to collect the questions. Stuart, the stage is all yours, and thank you for joining us today.
Thank you very much, and it's a real pleasure to be giving this talk to SONA. I have actually attended one SONA meeting in person, and it was a lot of fun and a very vibrant society, clearly. So just a couple of slides about Newcastle, just to say where we come from. This red dot here is Newcastle in the UK, up in the north of England, almost into Scotland, in fact, so far north. And Newcastle is a city known for its industrial heritage. You might have heard the phrase coals to Newcastle. We were a major coal mining centre until not many years ago, also a shipbuilding centre, and that we have this amazing quayside in our city with a famous for its number of bridges across the River Tyne. Medical school in Newcastle is very research intensive, and neuroscience is very strong in Newcastle. We have a lot of neuroscientists all occupying this building at the front of our medical school. And I think the two things that are going to be important in this talk to think about when we think about the neuroscience research in Newcastle, first of all, we are a major primate centre, as was mentioned in the introduction there. We're one of the two big universities carrying out research into non-human primates, macaque monkeys, and that's a very important model, I think, in motor control, which is what I'm interested in. The second thing is that we are a medical school and we have very close links to the local hospital, the Royal Victoria Infirmary. I can actually go from my primate lab with the animals through to the front entrance here of the Royal Victoria Infirmary without ever going outside. It's a seamless one building transition. And I think that really underlines the nature of the talk today. It's going to be a lot of basic neuroscience, a lot of it worked out in animals, mainly macaque monkeys, but thinking about the translational possibilities and how we can take that through to patients. Movement control research in Newcastle is very strong. We've got 6 research groups, 6 principal investigators shown here, all of whom have their own particular interests and research carried out by quite vibrant research groups into motor control. So to go on then with the plan for the talk today, as the title says, I'm going to be talking about portable devices and how we can use these in neuroscience research and in healthcare. And I'm going to be talking about 2 use cases, I suppose, 2 case studies. One is to think about stroke rehabilitation, and that's really a treatment. That's a way of actually improving the situation for these people. And the second is to think about diagnosis of movement disorders, and I'm going to be focusing mainly on Parkinson's disease. And for each part of the talk, although they might seem quite disparate topics, quite different ideas, there's the same basic plan. I'm going to be talking about the basic neuroscience underpinnings of the work, and that's really important. This is a neuroscience talk. Okay, this is not a technology talk. This is a neuroscience talk. So I'm going to be talking about neuroscience for most of the time, because we need to understand that neuroscience to understand how we might exploit it for clinical benefit. So then I'm going to be talking about technology and about some clinical proof of concept to show that some of these ideas actually work and could be used to benefit healthcare. So without more ado, let's dive into the very first topic, which is to think about control of a hand. Now, the hand is a uniquely primate acquisition, and there are some pictures on the right there of things that humans do with their hands and involve very high manual dexterity, so typing, shuffling a pack of cards, knitting, and performing surgery. All really skilled movements. And it was Kuipers, Hans Kuipers, who first introduced the idea that this skilled hand movement might have an anatomical substrate in primates. And the picture here from one of his papers just illustrates ideas that really Kuipers was absolutely foundational in bringing forward. What this shows is the distribution of corticospinal terminals in the spinal cord. And they're plotted for different animals, from a possum, a cat, a rhesus monkey, and a chimpanzee. And what you can see is that there's this ventral march of termination patterns as we go across this phylogenetic species series, such that the terminals get more ventral as we go up, until eventually in the higher primates, some terminals actually end up in the most ventral part of the spinal cord, grey matter here, where they're terminating amongst the motor neurons themselves. In other words, motor neurons are directly controlled in primates by the corticospinal tract. And this led Kuipers to say, well, this must be the most important system for controlling the hand. And he's absolutely right, of course. This is the system that gives us manual dexterity. But it's not the whole story. And I think the rest of the talk is really going to be talking about how this is not quite the entire story. And we can see that from just some older recordings from Muir and Lemon here. What they did was to record from a corticospinal neuron when monkeys did a precision grip and a power grip. And they found the corticospinal neurons modulated their discharge. This is just firing rate here versus time. When the monkey squeezed these levers, very nice modulation in discharge. But when the monkey used a power grip, a whole hand grasp, this cell hardly cared about this task at all. There's a hint there that corticospinal tract can't be the whole story for control of the hand. And indeed, there are multiple pathways that control muscles in the human and in other primates, and some of them are just shown here in a very schematic form. So we've got the motor neurons at the bottom here that project out to muscles from the spinal cord. The red pathways of the corticospinal tract directly projecting down from the cortex to the spinal cord, and you can see those direct connections here, those ventralmost terminations onto motor neurons. But there are also other connections from the corticospinal tract to interneurons, both higher up in the spinal cord and in the segmental level. And then finally, there's this extra pathway here, the reticulospinal tract, shown in green. And it also projects down to the spinal cord, and it has connections from the cortex. And if you look at the textbooks, they will tell you that the reticulospinal tract is much more involved in gross motor function, things like posture, locomotion, and balance, for example. And over the last few years, my laboratory has really been questioning and challenging that, and looking at the role of reticulospinal pathways in other movements, particularly control of the hand. And that's slightly heretical, perhaps, because we might think hand function is corticospinal. But we've shown in a series of papers here that, for example, the reticulospinal tract does make direct connections to motor neurons, even motor neurons involved in controlling intrinsic hand muscles. It makes connections to interneurons in the spinal cord, and many of those interneurons get shared input from both corticospinal and reticulospinal pathways. The reticulospinal tract is under the control of cortical projections and very widespread convergent inputs from multiple cortical areas here come down to reticulospinal neurons. And finally, reticulospinal cells themselves modulate their discharge very clearly with hand movements. If we ask a monkey to do even a very precise fine finger movement, then we can see cells in the reticular formation modulating very strongly. So all of these ideas really have changed our thinking a little bit about how the hand is controlled. Yes, of course, the corticospinal tract, very important, but there is this parallel pathway, reticulospinal tract, which is also involved in control of the primate hand. So does that matter? Well, it certainly matters when we think about motor control, but it might become particularly important if we start to think about how people can recover after damage to the corticospinal system. There are some numbers there, I won't read them out, for numbers of strokes and spinal cord injuries per year in the UK. And all of these are significant problems, significant contributors to disability. And very often these conditions involve damage to the corticospinal tract, and that leads to a flaccid paralysis of the hand initially, but then function recovers. And often there is some recovery, but the movements are not as good as they were before. And so we were interested, if you now believe that the reticulospinal tract is an alternative pathway alongside the corticospinal tract, then perhaps it could be what's providing some of that functional recovery after a lesion. And so we looked at that in our macaque monkeys. So just to give you some ballpark numbers, first of all, if we look at the relative importance of corticospinal and reticulospinal pathways, in a healthy animal, we found that there was roughly 5 times more input from the corticospinal tract to motor neurons than from the reticulospinal tract. But then we made lesions in our animals and we cut the corticospinal tract and they recovered. And after they recovered, we then measured the strength of these inputs again. And we found that there was a substantial strengthening of these inputs from reticulospinal tract to motor neurons, around 2.5 times increase. Furthermore, if we look up in the reticular formation itself, we then found that the cells here increased their firing rate by about 1.6 times. And if we multiply those two numbers together, we get about 4. So in very rough numbers, we're suggesting that after a corticospinal lesion here, reticulospinal tract can increase by 4 times the amount of drive it can provide to motor neurons. And you can see that might go some of the way towards helping recovery after this lesion. So that's spontaneous recovery. That's what happens when an animal or a human just recovers naturally after the damage, acute damage, such as you might see after a stroke or a spinal cord injury. But of course, the next question we asked is, could we help to improve recovery? Could we do something to strengthen those reticulospinal connections and thereby make recovery even better than it would be if we just left the animal or the human to recover naturally? And to think about this, we turn to a piece of basic neuroscience, which is called spike timing dependent plasticity. And this is the idea that synapses can be strengthened or weakened by being consistently activated in the right temporal order. And let me just explain that. So imagine we have one cell, the presynaptic neuron here, connecting to another cell, a postsynaptic neuron. If the presynaptic neuron is always activated just before the postsynaptic neuron, well, that's an indication that that synapse was actually helpful. It helped to depolarize the postsynaptic neuron and make it active. And so that synapse tends to get strengthened. Whereas if the presynaptic neuron fires just after the postsynaptic neuron, it was really not much use in helping that postsynaptic neuron to discharge. And so that synapse tends to get weakened. And so we end up with this classic spike timing dependent plasticity curve here. On the x-axis, we have time between activation of these two cells, and on the y-axis, change in synaptic strength. So if the presynaptic comes before the postsynaptic, we get long-term potentiation. And if the other way around, the postsynaptic before the presynaptic, we get long-term depression. And obviously one activation isn't enough. Here we need many, many consistent repetitions of this temporal order. And the timescale here is of the order of tens of milliseconds. So they need to be really quite precisely timed in order to see these effects. So we wondered, could we use this piece of basic neuroscience, basic biology, to change the strength of connections, the reticular formation? What do we need to do that? Well, we'd need, first of all, a way of activating the inputs to reticulospinal cells, the presynaptic cells. We'd need a way of firing reticulospinal cells, activating them. And that would give us control of both pre and postsynaptic neurons. And of course, we don't want to be putting electrodes in people's heads. We want a non-invasive way of doing it. And then finally, we need the ability to pair these two things together in very precisely timed ways. And that, in theory, would strengthen or weaken connections, depending on the timing. Well, how do we do that? Well, the first thing is we found a very straightforward way of activating inputs to reticulospinal cells, and that's electrical stimulation. We know there's very powerful somatosensory afferent input to these cells, and it's very easily activated by electrical stimulation. So here's an example. We're stimulating a peripheral nerve in one of my monkeys, and we're looking at a reticular neuron responding. There's a very clear burst of discharge here in the peristimulus time histogram in response to that electrical stimulus. This happened to be in a monkey. We were using an implanted electrode. But in a human, we can just use surface electrodes placed over a muscle or a nerve. We can activate things just the same way. So this is straightforward. The second thing we needed was a non-invasive way of activating reticulospinal cells. And it turns out that a good way of doing this is a loud click. This was a slightly serendipitous discovery we made. But here's an example of 3 sweeps of a cell within the reticular formation of the monkey. And it's responding to a click, an auditory click played through an earpiece. And you can see there's a little burst of action potentials in response to that. And here's a raster plot, successive trials, and we turn the intensity up and we get more and more action potentials generated in response to the click. We think that some of this response certainly goes through the cochlea. It's a genuine auditory response. Some of it, though, probably goes through the vestibular system, and essentially we're shaking the vestibular apparatus, and the loud click is capable of producing vestibular inputs to the reticular formation. And of course, we can do this very easily in a human, just with an earpiece that's delivering a clicking sound. So we came up with a proposal then for non-invasive, spike timing dependent plasticity of reticulospinal tract outputs. What we thought we'd do is to give an electrical stimulus to a muscle and a click to the earpiece. And we time these with a certain interval that we've worked out because we know some of the delays in these pathways, such that the input, the synaptic input, the EPSP from the stimulus to the muscle would arrive just before the little burst of action potentials produced by the click. And if we get the timing right, and we do this many times, this should lead to that EPSP growing. Whereas if we turn things around the other way, and we put the click before the electrical shock, now the reticular formation of cells will fire before the input occurs, and by the rules of spike timing dependent plasticity, that should decrease the size of that input. So long-term depression. So to test this idea, the third thing we need is a way of pairing these stimuli in a precisely timed way. And to do this, we developed this little box. We call it the wearable device. It's a small box about the size of a smartphone. It has a stimulus output port here with the ability to control intensity, and that will stimulate the muscles. It has a headphone output port, and it can all be charged up over a USB power supply. So it's battery powered, and it can be worn all day. And people can put this on. They can have electrical stimuli to their muscles here, a click sound through an earpiece, and they can wear it whilst just going about their everyday life. Does it work? Then, well, here's an example of an experiment we did to have a look at this and to see whether it might work. So the first study we did is we decided to use the long latency stretch reflex in biceps as our assessment to see if we could measure the strength of reticulospinal pathways, because we know that at least some of the contribution to that reflex is likely to be from the reticulospinal tract. So here's what we did. We had subjects come in, healthy human subjects, came into the lab around 9 o'clock in the morning. We sat them in this apparatus where they got very rapid perturbations of the elbow, and we could measure a stretch reflex in the biceps. That didn't take very long, took about 10, 20 minutes maybe. And then we fitted them up with the wearable device. So here this subject has electrodes attached to her biceps muscle. She's got an earpiece in her ear, and she's got a wearable device which she's got in this bag around her waist. And then the subjects went off for around 7 hours and carried out their normal daily activities. And after that time, they came back to the lab and they repeated the measurement of their biceps stretch reflex in the stretch apparatus shown there. And here are the results that we found. These are average sizes of the long latency stretch reflex measured across 15 subjects per condition. The circles are for before and the squares after the 7 hours of device wearing. These are the results for the paradigm where we put the biceps 10 milliseconds before the click. We saw an increase in the reflex. Here's click 25 milliseconds before biceps, and we saw a decrease in the reflex, exactly as we'd expect from spike timing dependent plasticity. Whereas if we just stimulated the biceps on its own, or the click on its own, we didn't produce any change in the reflex size. That's the control. So this looks really quite hopeful. It's exactly as we'd expect, and it really does suggest we are modifying these circuits through this very simple stimuli, very simple non-invasive stimuli delivered through a portable device. We've since done some more work, and you might say, well, how do you really know it's reticulospinal? And of course, that's very hard to do in humans. But we've just published a paper where we've looked in more detail at other measures, and here's one of them. This is the so-called start-react effect. We get subjects to make a simple reaction time response, move when you see the light flash, and we measure the reaction time, and we plot the cumulative distribution of reaction times here. The black line is a light combined with a quiet sound, and the red line is a light combined with a loud sound. And you can see we get a shortening of reaction times on average, and we call that the start-react effect. And there's evidence in the literature that's likely to be a good measure of reticulospinal pathway. Well, when we tested the start-react effect like this, just as the same way as before, one experiment in the morning, one late in the afternoon, and the person wears the wearable device in the intervening time, we find we can increase the start-react effect between these subjects. So that's a suggestion, again, that we really are producing plasticity in reticulospinal pathways. And then we've gone on to do a successful clinical trial, not in healthy human subjects now, but in stroke patients. So in this case, we targeted the wrist and finger extensor muscles, which tend to remain weak during spontaneous recovery. This was a study done at the Institute of Neurosciences in Calcutta in India. And we had stroke patients that were assigned to 3 different groups here. One group wore a paired device, which gave paired stimuli. One gave random stimulation between either, so the clicks and the stimuli to biceps were randomized, and one group just had no device at all for standard treatment. And we measured the action research arm test in these people over a period of 8 weeks. And for the first 4 weeks, they wore the wearable device for at least 3 hours a day. And we found that there was a significant increase in hand function in just the people who wore the device, which gave paired stimulation as shown there. So not only does this work in healthy human subjects for 1 day, but if subjects wear it for a longer period of time, we can produce functionally meaningful changes which actually have an impact on hand function. So to summarize part 1 then, I've shown you some functional anatomy. We talked about the role of the reticulospinal tract in hand function, and that it forms essentially a parallel pathway for hand control alongside the undoubtedly much more important corticospinal tract. And that parallel pathway comes into its own during recovery from lesion when it contributes to spontaneous recovery. And then if we give paired stimulation with a wearable device, we can induce plasticity in that reticulospinal tract, and that can actually improve function in patients. So the outcome of this is we've got a portable, really quite low-cost therapy which can improve the lives of people who've been unfortunate enough to suffer a lesion of their main corticospinal pathways, such as after stroke. Well, that's the first part of the talk then, that's been thinking about a way of treating a disease. The second part is to think about ways of diagnosing disease, and so portable diagnostics. And so now we're going to switch tack slightly, a rather different field of science, I think, within movement science. We're going to think about behavioural measures for going and stopping. I'm going to show you some ways in which we've managed to improve these estimates using mathematical Bayesian approaches to uncertainty. There's going to be some maths there, I'm afraid, so brace yourself for some equations. And then we've also managed to implement these measures in a portable form, and I'll show you how they could potentially help us to improve diagnosis of movement disorders. So first of all, let's think about the types of measures we're making here. And the first measure is a simple reaction time task. Imagine you're sitting at the traffic lights in your car and you have your foot on the brake and suddenly the light goes green. And so naturally you move your foot across to the accelerator as quickly as possible and you press and accelerate and go. It's a simple reaction time task. And if we look at the time of your response relative to the go cue, in a typical healthy subject, we'll see quite a range of distribution here. This is the distribution histogram of reaction time, and here is a cumulative probability plot. There's a few hundred milliseconds of variance in reaction time in healthy subjects. Now let's think about a slightly different situation. In this situation, again, you're sitting at the traffic lights waiting for it to go green, and it goes green. But then very shortly after the light goes green, something happens which means you really mustn't go. It could be a child running in front of your car. It could be an ambulance perhaps passing through the lights the other way with its blue light flashing. In our laboratory settings, when we test this, it's simply a red light appearing. And that tells you that you mustn't go. You must remain in position and should not respond. How do we think about this then? Well, obviously it's very difficult to measure what's happening in a situation where you don't respond. It's a much harder thing to measure than a simple reaction time. And we've used a model for thinking about this and understanding it that was first introduced by Logan and Cohen in 1984. And they called this the race model. And they said, let's imagine that there are two processes in your brain which are racing towards completion. The go cue occurs at a certain time, that's the green light, and it triggers the go process. And the go process rises up towards threshold, and when it hits threshold, you go, you respond. And the time it takes to do that is, of course, just the reaction time. But if the stop cue occurs, that triggers a stop process. And the stop process now rises up towards the threshold. And if the stop process hits threshold first, it will stop the reaction. Whereas if the stop process hits the threshold after the go process, then you will respond, even though that's not quite appropriate. There are two numbers we can look at here then. One's the stop signal delay. That's set by the experimenter. That's the time between the green light coming on and the red light, the go cue and the stop cue. And obviously, the longer that delay is, the more likely it is you're going to respond, even though you shouldn't really, because the stop cue has occurred. And the second thing here is the stop signal reaction time. That's the time it takes the nervous system to process that stop cue and abort the action. And that's an unobservable number typically. That's not something we can measure directly like the reaction time. But Logan and Cohen came up with a very clever way of actually measuring that. And they did as follows. What they did is to look at the proportion of times that the subject fails to stop and to combine that with the distribution of reaction times to a normal go cue. And they did that as follows. They said, so here's an example. Suppose here we have a stop signal delay of 100 milliseconds and the subject responds, so they fail to stop on 50% of the trials. Logan and Cohen said, well, let's just draw across on the cumulative distribution plot here of reaction times, draw across at the 0.5 point, drop down, and in this case, for this subject, that gets us to 370 milliseconds. So it's 370 milliseconds after the go cue that the subject is able to abort the movement. And that explains why on half the time, they're not able to abort the movement because half the time they've moved ahead of this point. We can then just subtract off the 100 millisecond stop signal delay here to get the stop signal reaction time of 270 milliseconds. Yeah? So just if we look back visually, you can remember that the go cue occurs just before the stop cue. This is the stop signal delay, and here's the stop signal reaction time. So if we take the time from the go cue to the point where you can stop, subtract the stop signal delay, we get the stop signal reaction time. That sounds quite straightforward. One of the problems, though, in the way that's been done up till now, and the thing that we've changed and innovated, is that it hasn't really thought very much about the accuracy of these measurements. So suppose the subject responds incorrectly on m out of n trials. What do we think their response probability would be? Well, the simple answer, the good guess, would be m divided by n. So if you respond 5 times out of 10, your response probability is a half. But it doesn't take account of how accurately we know that number. So if I know 50 out of 100 is clearly going to be a more accurate estimate than 1 out of 2. We can actually use some maths to look at this. We can think about what's the probability that we will see m responses out of n if the actual response probability is q. We can calculate that very straightforwardly. Q to the m, 1 minus q to the n minus m. Of course, that's not really quite what we want. What we really want is the other way around. We want to know what's the probability that the response probability is q, given that we've actually seen the subject respond inappropriately m times out of n. And in order to do that inversion between these two distributions, this gentleman comes to our aid. This is the Reverend Thomas Bayes, an 18th century clergyman and also mathematician, who devised Bayes' theorem. What Bayes' theorem does, it lets us invert these two so that we can calculate the probability of a certain distribution of q given the data based on the probability of the data given this number q. So when we use Bayes' theorem, we're able to come up with a new recipe for calculating stop signal reaction time. It's described there, and perhaps I won't go in the interest of time, I won't go into all the details of that. But it allows us to come up with a better estimate for stop signal reaction time. Normally we use multiple stop signal delays in these experiments, and the normal approach is just to average the different estimates that they give. But using this Bayesian approach, we can come up with a much more reliable way just by multiplying together these different probability distributions. And that comes up with what we call the optimal combination stop signal reaction time, or OC SSRT. And we think that's going to be a lot better as a measure for stop signal reaction time than the more traditional way that's been used, which we call average SSRT. So again, does it work? Well, in order to make these measurements, the great advantage of all the maths that I've shown you is actually it's quite straightforward. There's nothing very complicated or difficult in this maths. There's nothing that requires very great processing power. So we're able to implement those computations in a very small microcontroller, very low power microprocessor, and we've been able to put it into a portable box here. So the box has 2 LEDs, a red and a green one, a big push button that the subjects can use to respond. And after they've done about 15 minutes of testing, that's 3 blocks of 4 minutes with a minute's break in between, then we can come up with a result which gives the stop signal reaction time estimated from these measurements with quite high accuracy because we're using this Bayesian approach. Well, we validated this with healthy subjects. This shows the new measure, our new measure, optimal combination stop signal reaction time, plotted versus the more traditional way of measuring this, the average stop signal reaction time. They're well correlated. But furthermore, if we look at the test to retest reliability of this measure, we find it's much better with the optimal combination stop signal reaction time. So the variance from one measure to the next is much reduced compared with the average. And in fact, the interclass correlation coefficient is so high for optimal combination stop signal reaction time, and it's quite low for the average stop signal reaction time. And in fact, it looks like most of the variability in this correlation on the left is probably driven by the noise in the more traditional approach. So here we have a better way of measuring the neuroscience of stopping. Well, this is clinically relevant because several neuroscience disorders, several movement disorders seem to involve abnormal response inhibition. And we know that stop signal reaction time depends on the circuit of inferior frontal cortex, pre-SMA, the subthalamic nucleus, and the globus pallidus internal segment. But unfortunately, very few studies in movement disorders have used SSRT, which is much more theoretically attractive than other ways of doing things, just because it tends to give such variable measures and long recordings are needed. So we wondered if our improved measure, using this optimal combination SSRT, could be more useful as a diagnostic measure than some of the things that have been done before. And indeed they can. So here's a plot of mean stop signal reaction time and ordinary simple reaction time for healthy human subjects in white bars, and for 3 different types of movement disorders, so cervical dystonia, Parkinson's disease, and writer's cramp. And in all cases, the patients showed a slowing of reaction time and stop signal reaction time. Furthermore, if we look at Parkinson's patients and we take Parkinson's patients before and after they've taken their dopaminergic medication, we find that they get better on these 2 measures after they've taken their medication. So there's an improvement by a treatment which we all know works with Parkinson's patients. Interestingly, these 2 measures of reaction time and stop signal reaction time seem to reflect different aspects of the pathology, because in both healthy people and also in Parkinson's patients, there's no correlation between them. So we're not just measuring the same thing with these 2 measures. Probably reaction time is measuring bradykinesia and slowing of movement that we know is a feature of Parkinson's disease, and stop signal reaction time is measuring response inhibition. These are 2 different aspects of the same pathology. SSRT doesn't seem to correlate with the severity of disease, interestingly, in Parkinson's disease. It's more an on-off, either with or without Parkinson's. They have a slowing or not a slowing, but doesn't really progress with more progressive disease. And finally, it's all very well to say that these people are different between healthy people and Parkinson's patients. Does it actually have any utility in diagnosis? Well, actually, if we plot the cumulative distribution here of the OC-SSRT for healthy people and for patients, you can see the curves are really quite well separated, such that we can put a threshold at a particular point and we can say people to the left of that line are likely to be healthy and those to the right of that line are likely to have Parkinson's disease. Of course, the results will depend on very much on where we put the threshold, but we can move our threshold all the way across these curves and plot something called the receiver operating characteristic. So this is the true positive rate, the number of times we correctly detect Parkinson's disease, versus the false positive rate, the number of times we say that somebody who's healthy has got Parkinson's disease. And these will depend on where we place this threshold, but we end up with a plot that looks like this for many different thresholds. And the dotted line shows what this plot would look like if these 2 measures were completely the same in these populations. And you can see we're very comfortably away from that line. And typically in these sorts of curves, people will measure the area under this curve as a metric for how good this might be as a diagnosis. And we find that area under the curve is 0.95, which is significantly different from the 0.5 we'd expect if the measures were just performing at chance level. So these do really seem to be quite effective measures for diagnosing and separating healthy people from those with Parkinson's disease. Furthermore, we've looked at the relationship with mild cognitive impairment. So here's a plot of reaction time and OC-SSRT versus the score of the patient on the Montreal Cognitive Assessment. So 30 means completely normal and lower numbers means cognitively impaired. The blue lines are for healthy subjects to give you the normal range. The black points are for Parkinson's patients and the red ones are for those with Alzheimer's disease. The shaded regions are those that are said to relate to patients with these disorders that have mild cognitive impairment. And what's really exciting is that for the people with mild cognitive impairment with Alzheimer's, there's no change in either of these 2 measures. But for the people with Parkinson's disease, even though they have higher scores on the MOCA, they have an impaired stop signal reaction time and reaction time. And in fact, for the Parkinson's patients, even those who are not sufficiently impaired on MOCA to be called mildly cognitively impaired still have an impairment on stop signal reaction time. So this really does look very promising as a way of dissociating people with mild cognitive impairment who have a Lewy body disease, so Parkinson's disease, versus those who have Alzheimer's disease. An important diagnosis to make because treatments for those 2 disorders will of course be very different. Well, you might be sitting there when I said that we'd used a small portable box with a microcontroller to deliver these stimuli. You might be sitting there thinking, well, why doesn't he just use a phone app? Well, we have now developed a phone app for this. Actually, that's not trivial, and I think it's important to say that. There are some really big technical issues in doing that, in getting accurate measurements of things like reaction time using something as simple as a smartphone, but we've managed to solve those issues. And we've now got an app that does work on any Android phone. It gives the red and green cues via the phone screen and the response via the touch screen. And it just emails the results to the investigator. And I think this is quite an exciting opportunity, and this is really why I wanted to give this talk to SONA and to talk about the opportunity of developing an app like this into something which can involve further measures using some of the sensors that smartphones have, which could create perhaps a unified diagnostic system which could include multiple measures. And just one example of that, which I'd like to show you just to finish off, is to think about video analysis. Now, of course, people interested in movement disorders are very interested in looking at human movement and quantifying that. And typically in a laboratory setting, that involves the sort of systems we've got here. So we might have an instrumented mat here to measure gait, or we might have a large room with multiple motion capture cameras. And then subjects will wear these sorts of reflective markers as they move around the room, and we can capture their motion. And of course, it's a technology that's not just used by scientists, it's also used in the movie industry. So it allows movie makers to convert actors' movements and to render them onto animatronic figures, such as in this movie Avatar. But this is all hugely expensive and very complicated and quite impractical for anything other than the most specialist centres. But just recently, we've been experimenting with using much simpler videos taken on either webcams or smartphones and processing them through algorithms that use deep learning to work out where the different parts of the body are. So in this case, you can see we've been able to place little markers on the hand, the wrist, the elbow, the shoulder, and the neck, and we're able to track that in real time from these videos. So this offers the opportunity of making really quite quantitative measurements of human movements from something as simple as a smartphone. So to summarise part 2 then, I've shown you some basic neuroscience, the neuroscience of stopping, and a principled, analytically rigorous way to assess stopping using the race model. And I've shown you some improvements to that we've made using Bayesian statistics and portable electronics so that we can decrease the variability and make it a more useful measure. And this has clinical advantages because it seems to be able to measure something independently from reaction time, it's prolonged in Parkinson's, and it's effective in mild cognitive impairment in Parkinson's, but not in Alzheimer's. And conversion of this to an app and incorporation perhaps of other video-based measures could really produce a very powerful diagnostic tool. So the overall summary then, I've shown you 2 examples, one of treatment, one of diagnosis, of using portable devices and now an app to aid treatment and diagnosis. And I think there are real common themes here. This is absolutely driven by recent advances in technology, but it has a very strong neuroscience underpinning, and I think that's essential. If you go onto the App Store, the Android or the Apple App Store, you'll find lots of examples, for example, of apps that claim to help you to sleep. They really have no scientific underpinning at all. It's important we're very careful about not just adopting engineering solutions which have no underlying basis in the science and neuroscience. But I think these sort of approaches could really drive a new way to deliver healthcare, and they're particularly interesting in a developing nation setting. And I just want to draw the analogy perhaps with how technology has developed a bit. In the 1960s, if you talked about technology, you'd be meaning something like this, a mainframe computer. These were hugely expensive. They had to inhabit purpose-built spaces, clean, with lots of cooling. They consumed large amounts of electricity. And you couldn't use this unless you were highly trained. It had to be highly trained staff, and it was very complicated to maintain. And as a consequence, computers didn't do very much. There weren't many problems that were thought sufficiently important to be solved by a computer. And perhaps the analogy in a healthcare setting there would be the hospital. Hospitals, purpose-built spaces, very expensive, lots of facilities, lots of trained staff, and only used for the most severe illness. By the time we got to the 1980s, technology had evolved and we had personal computers. A wonderful advance. These were now cheap enough to be in every office. But they still needed a lot of expertise to use. They had rather limited computing power, and they consumed sufficient electricity that they had to be plugged into the mains. They weren't portable. And indeed, leaving your personal computer on overnight could rack up quite an interesting electricity bill by the morning. But now we've moved on, and perhaps the analogy, to think about healthcare here would be the family doctor's practice, the local family doctor's surgery. But now we've moved on with current technology in a real revolution. Laptops, smartphones, tablets are now cheap enough to be really everywhere. If you take a bus ride in almost every country in the world, your fellow passengers will be looking at their phones. They're easy to use. There's been a huge change in usability, such that most 5-year-olds can use these sorts of devices. Most grandparents now can use them and are happily making video calls to their grandchildren. They use much lower power consumption, and that makes it feasible to power them from small batteries, so they're portable. And increasingly, they're incorporating sensors like smartphone cameras and accelerometers, which could be used in the sort of applications I've talked about. And finally, and absolutely critically, of course, these are internet linked. And that has a number of key things that really is a game changer. The first is it means that expertise no longer has to come to you. It can be downloaded either by social networking or by forums, for example. You can just Google something. Maintenance is no longer a man coming with a toolbox. Maintenance can be a software update. And that's really important when things have to be used out in the field. And finally, cloud computing means if there's a problem which is too difficult in terms of computer power to solve on your own device, you can simply upload it to a server somewhere remotely, and it can be solved on that server using much more computing. And an example for example, would be the video analysis I showed earlier. That might work on very cheap videos taken on smartphones, but the analysis is really very processor intensive. And to do that requires quite a powerful computer. But that's not a problem because we can just upload the videos to a server and the server can do the analysis for us. And so this really offers the opportunity, I think, of taking healthcare away from these highly centralized settings right out into the community, such that it can be used by individuals in their homes, in their families, in a much more widespread way. And that could be a real transformative technology, particularly, I think, in a developing nation setting. So my outlook then is I think modern technology does offer this chance to move medical care away from the centralized model. And I've shown you 2 examples of that. A cheap portable device, which could be a useful way of enhancing rehabilitation after stroke, and ways of diagnosing neurological degenerative diseases using portable devices and now smartphones, which could help us enhance that diagnosis. And I'll just finish by thanking all the people in my group and my collaborators in other universities who've contributed to this work, and our funders, of course, at the bottom. Without whom none of this would happen. And final slide, I just want to have 2 pieces of advertising. The first is that, as was mentioned at the start, I'm very much interested in looking for collaborators, those working in Africa, who might be interested in applying some of these methods in the context of their own healthcare settings and thinking about how we could use these methods to make a difference. And a final shameless piece of advertising. I am part of a network, the Into Primate Brains Network, which currently has 15 funded PhD positions in primate neurophysiology in multiple European labs, including in Newcastle. And we're currently recruiting, so we're currently looking for 15 PhD students, fully funded positions, might be of interest to some listening in. I would just emphasize though, please think very carefully about your skill set before applying, because we are looking for very strong computational and mathematical skills, not necessarily some of the wet biological skills. But clearly, these are neuroscience PhDs. You have to be very interested in the neuroscience. Okay. So with that piece of advertising, I'll finish, and thank you very much.
Thank you very much, Stuart. That was a very interesting talk. I guess to start with the collaboration issue, perhaps one of the things that could be done is if you could just maybe send out a brief info on the specific areas that you want. And I think this can be sent out to the SONA to all SONA associated organizations across Africa. I think that would serve as a more advertising platform, because maybe a lot of people have not had the opportunity to come and attend this talk or to watch it later on, but through the email, we could put the link to the talk as well as some of those things, because I think there is clearly a way of collaboration that can emerge from this.
Okay, thank you very much. That would be very useful. And I think it's important to say that I have an open mind about these things. I have ideas about what might be useful and might work. I have almost zero experience of healthcare in Africa. And so one of the things I'm looking for is advice and help from people who think what would really make a difference.
Yeah, absolutely.
Turn around to me and say some of these things are not that useful, and that's fine. I need to hear that, and I need to hear what would make a difference. And I think that's why it's not a one-way thing. We have to have a really collaborative discussion about what would be useful, what could we do, what would be practical, and how can we do it together.
Yeah, absolutely. I mean, hopefully I'll go back to the Facebook as well to check if there are any questions. And for people watching from here, if you could send out questions. But in the meantime, I was wondering, I didn't catch it very well. Did you show that the reaction time is kind of more reliable for movement disorders, but not Alzheimer's or dementia? Was it, am I correct?
It wasn't so much reliability, it was just the size of the reaction time. So just to go back and show you, there's the plot. So so this is reaction time, this is stop signal reaction time. This is the healthy range, the blue, and it is elevated in the Parkinson's patients. It's elevated in the Alzheimer's patients when they become very severely impaired.
I see.
But with mild cognitive impairment, the shaded region, the Alzheimer's patients have actually normal reaction times, normal stop signal reaction times, where the Parkinson's patients, even though they're doing better on the mocker, they have impaired values here. So if you come to me with a person and you say, this is a person with mild cognitive impairment, do they have Alzheimer's, do they have Parkinson's? I can make this very simple measure and tell the difference between the two groups. Would be the
yeah, which is good. I mean, this is quite useful because even from the angle of diagnosing or differentiating between these different disorders, especially, for example, even for Alzheimer's, I do know that we don't have the infrastructure. The infrastructure for diagnosis is quite weak, and oftentimes it is difficult to tell whether it is due to Alzheimer's or is it just aging, or is it due to some dementia induced by some other factors. So I can see this being very, very helpful in that setting. Having said that, so in these experiments, what was the sample size, and were you able to check different forms of, let's say, Alzheimer's, MCA, mild cognitive impairment, and maybe some form of impairment due to, say, trauma or something like that?
No, I mean, we haven't done. That's the sort of workup that's needed, clearly, and I think that's one of the directions we need to go in, is to look at all of that with much larger samples. This is all work, I should say, and I perhaps didn't give the shout out to this as I should have done. This is all work done in Calcutta, Institute of Neurosciences, Calcutta, which is an amazing tertiary referral hospital with quite large numbers of patients. But nevertheless, it's a rather specialist setting.
Yeah, absolutely.
Any tertiary referral neurology hospital is not going to be looking at the sorts of patients that you might be most interested in a community setting, the people with mild disease, the people who haven't even thought of going to a doctor yet. And so I think that's where we need the collaborative input here, is to think about trying to get to some of those people. And absolutely increasing sample size, stratifying them, thinking about all the different diagnostic options, that would be really important.
Yeah. All right. So we do have a question. So in terms of setting up some this kind of thing, how was the cost implication? I mean, is there any cost implication in terms of if people want to set up something like this? I mean, what are the nitty-gritties that researchers need to know? This is from Facebook.
Yeah, so I think the cost implications, clearly, the devices I showed for stroke, they would have to be made. That's not a smartphone device. It's giving electrical stimulation. It's a relatively cheap device to make if we made it in even small quantities. So I think once we start mass producing a device like that, the cost would come down to something very, very low. I mean, I'm thinking maybe if it were really mass produced in an African setting, we might be thinking about 20, 50 pounds perhaps, and it's a reusable device, so people don't need to use it very long.
Yeah.
In terms of the smartphone app, we need some more development time. I think it's really about more research time needs to go into this to develop something that's usable to get feedback. And I think we're at the stage of not just producing something as a toy. We need to be producing something that's usable, and that needs all the usability and feedback from end users. So I think that's where the cost is going to come in, is doing that sort of work. Ultimately, once we've produced it, the cost, I'd like to think, would be zero, because we could just make this app freely available, people could use it. So at the moment, I think it's a research project to try and develop this into a usable tool. That's probably going to need some funding, and hopefully one of the ideas we have is we'd like to apply for some joint funding with African collaborators.
So I mean, yeah, it does a really nice kind of ending. So I was thinking, so if that's the case, what range of things are you looking at? Because certainly it can be applied to so many different scenarios. So if for people that would come back and watch this or something, what kind of areas do you think, you know, would be exciting for you as well in terms of looking at these collaborations? And especially why I'm asking this is because yes, we do have quite an aging population, but at the same time, the youth population is also very high. So maybe it's good to not limit it just to say aging related conditions, but maybe there could be some application for this in community settings for even young onset forms of conditions.
Absolutely, and I'd be interested to hear what you think would be useful. I mean, obviously, I'm interested in movement control, movement disorders. That's where our expertise lies, and so that's where very much we have focused, and I think where we would sensibly focus for the immediate future. I'm sure that there could be a place for some of these things in, for example, epilepsy, for example, a whole range of other neurological disorders, which we can think about. That's not my expertise, and it's probably not where I would want to go with it immediately. But for example, video analysis. What's the first thing a neurologist will ask you if you say you think you might have epilepsy? They'll ask you and your family to describe what the seizures look like, the semiology of the seizure. And very often, the neurologist is delighted if the family can show a video of what the seizure looks like, because that makes the diagnosis so much easier. Well, you know, we could be thinking about doing automatic analysis of video, putting that out. So that would certainly be possible. It's not my expertise, but it's certainly one of the areas you could think of going.
Yeah, I agree.
Childhood development disorders as well, childhood neurology, huge opportunities there, I think, for doing something similar.
Yeah, I think it should be, I mean, in the interest of time, we probably have to stop here, but I think it should be super cool if you are able to just send out something which we can put together with this link to send out to everyone. I mean, the Secretary General of the Society of Neurosciences of Africa is also here with us. I'm pretty sure he'll be happy to do that. But this is such an exciting thing. And like I said before the talk, there are some discussions, especially in relation to dementia. I'll hopefully follow up with that to see where it goes. Yeah, I mean, thank you very much, Stuart, for a fantastic talk. And I agree this is highly, highly relevant for the African setting. And hopefully this will trigger some discussion and collaboration to take this work forward.
Great. Okay. Well, thank you very much. Thank you all for listening and for hosting me. Thank you.
Thank you.